Okay, so welcome to this class on neuroscience of human movement. This is part 14 of our discussion on primary motor cortex. We are almost done with our discussion on primary motor cortex. In this class and in the next class, we will be summarizing what we have learnt in the previous classes. So, in a way this can be called as summary part 1. So, what we have learnt is just so much in the past 12-13 lectures that the summary itself requires 2 parts. So, this is part 1 of summary or part 14 of the discussion, whichever way you want it. So, what did we learn in the past so many classes? First and foremost, historically, localization of cerebral function started with around the same time by multiple colleagues, multiple people or multiple stalwarts. Paul Broca, his suggestion that the speech production was localized in a particular area, right, that is now called as the Broca's area, right. Then for speech perception, Wernicke claimed that there is a particular region within the brain where perception of speech happens, which is now called as the Wernicke's area, right. Around the same time, John Hewlings Jackson made a claim that would now be considered controversial for that time, right. He made this bold claim that there is this particular region that is just rostral or just anterior to the central sulcus, what is now called as the precentral gyrus, has a crucial role, a significant role in movement. Now, that so this discrete region just anterior to the central sulcus is now called as the primary motor cortex, but this claim was made in the middle of the 19th century. How did he make this claim? He was actually a physician treating patients with seizures. So, based on how these patients behaved, the movements that these patients made, it seemed to him that, or it appeared to him that the seizures, the way the seizures proceeded, based on that he came to the conclusion that these regions that control neighboring regions of the body must also have a neighboring control centers. This led to one thing led to another, this led to further studies, and finally he made this bold claim, or what would have been considered revolutionary at that time, that the region in the cerebral cortex that is just rostral to the central sulcus or the precentral gyrus has a crucial role in movements. This is now called as M1, is it not, or primary motor cortex and premotor cortex now. And in the 1870s, at around the same time, David Ferrier in England and Fritsch and Hitzig in Berlin around the same time studied dogs, and they found electrical stimulation of the cortex evokes movements on the contralateral side of the body. Now, that means that when I stimulate a particular area in the brain, movements are produced on the contralateral side of the body, means that there must be a relationship between that site of stimulation and the movement that is being produced. Let us remember that this is 1870. This is about 150 years ago. So, for the good old days, that was a very good progress, that was a substantial contribution, right. And further, in the 1900s, in the early 1900s, Alfred Campbell and Corbinian Brodmann classified different brain areas using approaches of cytoarchitecture or cytoarchitectonic procedures. And what they found was that the area that is just anterior to the central sulcus or rostral to the central sulcus was agranular. It lacked the granular structure that is typically found in other cerebral areas. So, something was different about this area, and let us note this is from the viewpoint of cytoarchitecture, not function. from the viewpoint of cytoarchitecture alone, right. So, based on that they found that these areas must somehow have similar function different from other areas, right. So, once again let us remember this is the early 1900s, more than 100 years ago, right. And in the meanwhile Sherrington, using his own classical approaches, and he came up with this idea that the motor cortex in great apes are organized in a certain way using electrical stimulation, right. So, basically the first or a first precursor to formal study of the organization of the motor cortex was of course earlier done by Fritz and Hitzig, but a proper step by step approach, a cleaner map and the organization was generated by this stalwart Charles Sherrington, and he studied this in great apes, right. And this work was continued later by Clinton Woolsey. What he did once again is a stimulation. So, he mapped the brain and explored the working of touch, hearing, vision and among other things. What he also found was that, so there are areas other than the primary motor cortex or within the premotor area, a particular area which when stimulated also produces movements. This they called as supplementary motor area. Let us remember that later this was further classified as supplementary motor area proper. We have discussed this in one of the previous classes, this, right, this is, and pre supplementary motor area, and then the other premotor areas are classified as dorsal premotor area, ventral premotor area, pre-dorsal premotor area, and the ventral premotor area itself is further divided into two areas. We have discussed this previously. And let us remember this work by Clinton Woolsey was performed on animals, okay. And later Wilder Penfield in the 1930s, 40s and 1950s studied the organization of motor cortex in humans. How can one do that? Well, obviously these were patients of epilepsy who required neurosurgery, and Wilder Penfield was this neurosurgeon. So, this is data from hundreds of patients, and during the surgery he stimulated certain parts within M1, and he found that there was disproportionate representation of hand, fingers and the face when compared with the rest of the body. This he called as the little man within the brain or the homunculus. Now, we know that this little man has some importance, not completely important. So, this little man of some importance, okay, or some significance, not huge significance. So, it is tempting to immediately attribute that specific area within M1 directly controls this finger or this particular region, and no other area controls that. Actually, Wilder Penfield never made any such claims. He just said that the total representation of the total representation, there is disproportionate representation of the hand, fingers and the face. So, that is the claim that is made. He was cautious enough to avoid such claims, right, but it would be tempting for students to come up with exaggeration. So, one should avoid that actively. And then later in 1950s, Herbert Jasper came up with this approach where you can record from live behaving animals. So, now that is revolutionary, because these animals are behaving as they would otherwise do. So, they are not or at least they are live and moving, they are not anesthetized, they are not undergoing surgery for example. And then when they are making movements, you could record the activity in different parts of the brain, right. So, this is microelectrode recording. Let us also remember technology that is required for these type of approaches also developed at around the same time in the post war period. So, there are multiple factors that has led to the development and an improvement of knowledge in this field. Then in the 1960s, A.D. Watts recorded from live behaving monkeys and he suggested that single neurons in motor cortex area discharge during movements of contralateral part of the body. So, once again let us remember this is data from individual neurons. So, he correlated activity of individual neurons with movements made by specific parts of the body, right. Further, what was also found that found by Edwards was that he found that there was a relationship or a good correlation between the amplitude of the force that was produced and the activity that was observed in specific neurons or in specific neurons within M1. Following up on that work, Epfetz found that in isometric force production, so force production can happen in multiple ways. When the object is not actually moving, you could still push on the object, on a mechanically fixed object, in multiple directions, for example. So, this kind of force production is called as isometric force production. We have explained what is isometric force production or isometric regime in the previous classes, and Epfetz found that the level and direction of isometric force also resembles how movement would have happened. So, this is similar to the actual movements and the actual forces produced during movement. So, in the isometric case there is no actual movement, let us remember that. So, that is the contribution of Epfetz. And in the 1980s a revolutionary idea that populations of neurons together encode movement features or movement related features. So, this is in particular what was encoded in a center out reaching task. For example, the direction of movement was found to be encoded in the activity of multiple neurons. What is the difference between this work and Edwards' work? Edwards studied activity of single neurons versus this. Here summation of activity of multiple neurons give rise to or give some insight into what is the actual movement that is being made or what is the aspect of movement that is being made. For example, the movement direction in this case. And Kalaska used a similar approach, and what he found was that the direction of reaching movements against external loads, right, against external loads also can be predicted by studying populations of neurons. Now, what this means is this external load could either assist the movement that is being performed or it could resist the movement that is being performed. Depending on whether the movement whether the movement is against the external load or along the external load, the activity or the population activity of these neurons changed and the directions changed depending on that. That is the result from Kalaska's work. Then the work of Mark Schieber in the 1990s is related to the somatotopy in the M1 hand area. Brilliant work studying or a crucial work in this field, studying the contribution of individual neurons or groups of neurons within the M1 region. What was found was that neurons that control movements of individual digits were not necessarily restricted to a small area, but rather were present in multiple areas, not necessarily in one area. They were present in relatively large, they were distributed in a relatively large area of M1. Also, what was found was that neurons within a region control not just one digit, sometimes control more than one digit, several digits at the same time. So, what was found was that there was not a region to region, a region to effector one to one relationship, but the relationship that was found was rather mosaic. There was both convergence and divergence. This theme I will continue in some of the later sections. Also, I will discuss this topic of convergence and divergence. Okay. And in the meanwhile, there was this work of Lawrence and Kuipers where they studied lesions. What they found was that lesions, pyramidal tract lesions on monkeys resulted in devastating effect of loss of fine control and claw like movement. So, objects that were picked earlier with the precise movements of the index finger and the thumb were now picked with the whole hand movements or claw like movements, right. And later there was this work of Peter Strick, right, where he studied pre and post lesion and showed that pre lesion the movement was diagonal as expected, and suppose that was the target and that is the start, right. If that is a movement that needs to be made, that is smooth movement before the lesion. After the lesion, the movement was like that. Remember this picture from one of the previous classes, right. So, this is post and the red is pre lesion, okay, that is pre. Then comes the revolutionary extraordinary contributions from Mike Merzenig's group and his trainee Randy Nudo, talking about plasticity. This concept is revolutionary, has important crucial implications for rehabilitation. So, what they found was that there was use dependent or activity dependent plasticity. So, as people learn there is reorganization. As they unlearn and forget, there is reorganization. Let us remember one thing. So, I will briefly summarize this situation. There is a state 1 before learning. This is the state of the brain before learning, and then the monkey or the human learns a particular task. Then they go to state 2. Then let us somehow assume that they have completely forgotten or unlearnt that task. You would expect that the brain would go back to state 1, but that does not happen. Usually what happens, that there is a new state that is achieved, that is state 3. This is after unlearning. This means that the brain is continuously remodeling itself. So, there is dynamic alterations in the brain structure as we go along. Then the work of Andy Schwartz, where activity of a population of motor neurons can predict the actual trajectories of movements. Let us remember this case where there was a spiral that had to be made from inside to the out or from out to the in, like that, for example. Remember that case. From these velocities, instantaneous velocities can be extracted, and population vectors can also be used to rebuild this trajectory, and the predicted trajectory from population vectors closely resemble the actual trajectories. This was the study that we discussed, that was Moran and Schwartz, right. Please revise this as we go along. I am just briefly touching upon what was already taught, right. Then the crucial contribution of Michael Graziano. So, what was found? Long lasting electrical stimulation, very different from other classical neurostimulation methods. A few milliseconds was the time that is used in classical methods. In this case, long lasting electrical stimulation, several hundreds of milliseconds, produce well coordinated smooth movements. We said this, defensive postures or hand to mouth movements. Let us remember, a hand to mouth movement involves movement at multiple joints, and that means control or finely coordinated temporal and spatial control of multiple muscles. This was achieved by long lasting electrical stimulation, a revolutionary idea. And continuing along, what was also found was that neuronal activity depends on the direction of the force. This is Klaska's work, and then Peter Strick's work, more contribution from Professor Strick. What he found was that there was divergent connections from corticomotor neurons to the spinal motor neurons. So, what this means is that, so if there is a corticomotor neuron here, there is a, suppose what does this refer to? This refers to the spinal cord. Obviously, I am drawing the classical spinal cord picture. This could innervate multiple areas. So, this corticomotor neurons can innervate multiple areas, and also remember one area can receive inputs from multiple neurons, right. So, what this means is that there is a convergence of so, when you look at it from the spinal cord point of view, at a particular point several motor neuronal inputs may converge, and from the viewpoint of the motor cortex, regions are from a relatively small region within the motor cortex, inputs may go to several areas or divergence. So, there is both convergence and divergence in these connections. Okay. This also we have discussed in the previous classes in relatively good detail. So, please do go through that. And more recently in the 2000s, what was found was motor learning, or in particular this is learning to respond to perturbations, right, to specific perturbations, and recording from neurons. It was found that the change in the directional tuning of learning was only specific neurons were making changes to directional tuning curves in specific directions, but not other neurons. There were other neurons that were responsible during the washout period, there were some neurons that were responsible during the early learning period or early change period, and there were some neurons whose activity did not change, right. So, this is the work of Beatsy that we discussed in one of the previous classes, and then also the work of Darcy Griffin, where he said that activity of specific M1 neurons can be correlated with specific patterns of muscle activity of specific muscles. So, this is a very brief summary of the work that we have discussed in the past several classes. What is missed in this summary? Actually, a lot is missed in this summary. Knowledge in this field has come through the work of several hundreds or if not thousands of researchers. I have only summarized the work of about 24 researchers here. So, the idea here is to use this opportunity to continue to learn. So, I have summarized what we have discussed in the previous classes. By no means this is justification of the actual contribution of various colleagues, and this is also not enough justification for the amount of knowledge that is there. This is just the beginning of learning. So, with this we end the discussion on primary motor cortex and studies of the primary motor cortex. Thank you very much for your attention.